Thank you, Eike. Is my sound okay?
Excellent.
Perfect. Okay, well, hello again, everybody. It's nice to see such a great discussion going. So it's my pleasure to introduce our next speaker, Qingyu Zhao, who is a faculty member at Stanford in the Department of Psychiatry and Behavioral Sciences. Working on the intersection between machine learning and neuroimaging and biomedical phenotypes associated with neuropsychiatric disorders. And he's going to talk about confounder-aware deep learning models for neuroimaging applications. Please go ahead.
Yes, thank you for the introduction. Can everybody see my screen, see my slides?
Yes.
Yes. Okay.
Yeah.
Okay. Let's get started then. Yes. I'm Qingyu. I'm an instructor in the Department of Psychiatry and Behavioral Sciences at Stanford University, and today I'll be talking about some of the approaches that we have developed for the past few years to control for confounding effects in deep learning models in the context of neuroimaging studies. So in those previous presentations, we heard a lot about the tremendous advancement brought by artificial intelligence and machine learning in different medical applications, and notably in radiology. And I'm going to switch context a little bit to talk about their applications in neuroimaging studies. It's certainly the case that machine learning is starting to revolutionize how neuroimaging studies are being performed. So in my lab, my collaborators and I aim to use machine learning or deep learning to aid computational neuroscience research. Essentially, we try to use unbiased and interpretable machine learning analysis to investigate biological, neuroimaging, and neuropsychological data in order to improve understanding of normal neurodevelopment across the lifespan, and also to help understanding the developmental course of some of the neuropsychological diseases and their interactions with daily behaviors. So before talking about machine learning and the concept of bias or fairness, I want to give a very brief overview of a typical workflow of neuroimaging studies so that you have a general picture of the analysis that was traditionally performed and why we need machine learning models as an alternative approach. So in order to investigate the effect of some neurological disease on the human brain, what you first do is that you first recruit a group of participants, some healthy individuals and some disease patients, and then start to take neuroimaging data from them. And then from these raw MRI scans, we apply a series of sophisticated pre-processing pipelines, which can basically segment the brain into different tissue types and a bunch of brain regions. And then you can extract features from each region. For example, you can measure some structural features, for example, the cortical thickness of gray matter regions, or diffusivity measures for each wet matter region, or functional connectivity between pairs of brain regions. And with these features, we can basically apply a hypothesis testing procedure based on a certain statistical model to each feature separately to see whether the disease can create a significant group difference or not. Well, this kind of analysis generated numerous amount of important findings about the brain, but we have to realize that the hypothesis testing procedure is basically a univariate analysis that does not account for the interrelationships across different features, and the analysis is basically based on group level inference that is confined to population level findings. So a recently emerging approach that I'm going to focus more on today is essentially to take the raw brain imaging data and feed it into a deep learning model to predict whether the subject belongs to the disease group or the control group. So this analysis generates subject level prediction, which can better stratify the heterogeneity under the population. And it also enables personalized diagnosis or treatment, which is a very important concept in precision medicine. And another advantage of this approach is that it's generally believed deep learning methods have higher sensitivity in identifying the disease effect, because it can better capture the complex interrelationships between the information that comes from different brain regions. And meanwhile, the disadvantage of deep learning, of course, is first of all, it's very data hungry. And even if you provide enough data, the advantage of high sensitivity in identifying the disease effect can sometimes backfire a little bit, in the sense that it can also be very sensitive in identifying the bias effect in the data set. So these two articles from Nature Medicine very well summarize the issue of having bias in current AI models and used in healthcare. And you heard of some of the talks in the previous presentations. Okay, so we have talked about the concept of bias and fairness in previous talks, including very nice overview talks and some of the specific cool methods for controlling or removing the bias. In fact, I believe a lot of those concepts can also be formulated in terms of confounding effects. This is a terminology commonly used in the context of neuroscience research. And so what are confounders and confounding effects? So confounders or confounding variables are essentially extraneous variables that can influence both the input and output variables. And some people call these independent and dependent variables depending on the context, but the upshot is that the confounders can distort the apparent relationship between the input and output so that it can create an spurious or distorted association between the two variables. So in the conventional statistical approaches, there are substantial efforts in modeling confounding effects. But I guess when it comes to machine learning, people start to overlook this issue a little bit. And as a result, currently there's no golden standard in for controlling confounding effects in machine or deep learning models. Actually, to give a more concrete example, let's think about a scenario where you want to use the model to predict the diagnosis label of a subject based on their gray matter volume, some information from the MRI data. And in this case, age can become a confounder because apparently age can affect both the gray matter volume and the diagnosis outcome. So when you grow older, your gray matter shrinks. This is a phenomenon called atrophy. And on the other hand, once you grow older, the likelihood of contracting a disease also gets higher. So without controlling for the aging effect, the model can simply learn the relationship between gray matter and age instead of learning the disease effect. In other words, the model will simply predict the subject's age instead of the diagnosis label. And there are several ways that are considered acceptable in dealing with confounders. So basically, you can categorize these approaches into two categories. And the first one is to remove the link between confounders and the prediction outcome, for example, by resampling the data set so that the disease group has the same range in confounder values as the control group. And there are several other approaches along the same line of thinking, for example, based on standardization or stratification, but I'll not go into details of those approaches because I want to focus more on how to remove the association between the confounders and the input data in this talk. And it turns out that in traditional machine learning algorithms, this is still manageable, still doable, because for traditional machine learning algorithms, you still have to extract manually defined measurements from the data set. So as long as you have these fixed deterministic features before running your machine learning analysis, you can always apply these traditional statistical models, for example, based on a general linear model, to find out the relationship between these features and the confounder value. And then you can residualize the confounding effects from each of these features so that the resulting features are considered as confounder free, which can be input to your machine learning model. But this approach basically becomes immediately infeasible in deep learning context because now you don't have features, so to say. And in deep learning, the idea is that you learn these features in an end-to-end fashion on the fly. So essentially just feed the raw MRI data into the model and you let the model automatically to decide which features to extract and how to extract them. So it becomes quite unclear how to optimally control for the confounding effects for this type of learning process. So the next part of this talk is going to be a little practical. I'm going to very briefly overview two approaches that we developed to handle confounding effects in neuroimaging studies. And the first one is this paper we published 2 years ago in Nature Communications. And this is also the time when a lot of new proposals were being made in the field of fair machine learning or trustworthy machine learning. And we were one of the first few to investigate the feasibility in the context of handling confounding effects in medical imaging studies. Okay, so the idea is that if you look at a typical convolutional neural network, it's usually the case that you will first use a bunch of convolutional layers to reduce the raw MRI data to a lower dimensional embedding or feature vector. So I call this component the feature extractor here. And then from the extracted features, a fully connected network will perform the final prediction of the diagnosis label, which I denote as the variable Y here. So the difference between these features and the features I mentioned earlier in conventional machine learning algorithms is that these features are learned dynamically, right? So they are not fixed or deterministic beforehand. So in order to prevent the prediction from being confounded by a confounding variable, let's say the variable C, we basically have to find a way to remove the confounding effects from these dynamic features. And the idea is that the natural idea is basically to use an adversarial type of training to learn the features that can predict the diagnosis label Y, while not being able to predict the confounder value C, so that we can ensure that the learned features only contain information related to the disease, but not related to the confounding information. And note that this part of not being able to predict the confounder values is achieved by adversarial training. I guess I'll not go into details of this procedure, but roughly speaking, in terms of the training objective, here I give a very high-level description, which is a min-max optimization that aims to lower the prediction loss for your disease classification and also increase the prediction loss for your confounder prediction. I do want to note that you'll be able to see a lot of similar formulations in the field of fair machine learning around that time. But what we did a little bit differently in this work was that we used Pearson correlation as the adversarial training loss, as opposed to commonly used the entropy-based loss. And this really allows us to handle continuous confounding variables such as age, in addition to only looking at or sex or ethnicity, those categorical variables. And another unique proposal in this framework is that we only train this confounder prediction component only in the control cohort in order to properly disentangle the confounding effects from the true disease effect. And it turns out that this practice is quite critical too in getting the desired outcome in our experiments. So we validated this approach in several medical imaging data sets, but here I'll just talk about one neuroimaging application, which is to use T1 weighted MRIs to classify healthy individuals from those with HIV infection. So here the HIV patients are significantly older than the controls, so age becomes a confounder in this study. So we propose to evaluate the effectiveness of this approach based on the accuracy measured on a so-called C-independent subset, which is essentially an unconfounded subset of the test data where patients and controls are matched with respect to their confounder values. So we can see here that on the whole data set, on the whole test set, the basic convolutional neural network achieved a 71% classification accuracy, but it was then reduced when you try to measure the accuracy on the unconfounded subset of the data. And this is not the case for our approach, which resulted in a consistently high accuracy on both the whole test set and also the unconfounded subset. And another piece of evidence I want to show here is that you can basically plot or visualize the prediction scores produced by the model for all the participants, where a 0 score essentially means that the model predicts the subject as a control and 1 as a diseased patient. And we can plot the distribution of these scores separately for young and for old participants. And we can see that for the regular convolutional neural network, both young controls and young HIV patients were more likely to be predicted as controls. And this indicates that the model was essentially confounded by age, because it thinks that as long as you are a young person, you should always be predicted as a healthy person. And this is not the case for our method, which says that for both young and old participants, the prediction should be mainly driven by the HIV diagnosis. And in fact, we can also look at the feature space learned by the models using testing projection. So basically each dot here in this plot corresponds to a features of a single participant projected onto the 2D plane. We can see that for the baseline approach, there's a clear age pattern in the feature space, in the sense that older participants are more likely to be clustered in this region, whereas if you look at our feature space, it's basically being impartial to age. And lastly, you can visualize the brain regions that are being used to drive the prediction. And you can see that for the baseline approach, a lot of brain regions are being involved in this process. And many regions, including, for example, the lateral ventricle, might have been just simply linked to the confounding aging effect. And on the other hand, if you look at our approach, it only used more confined regions to achieve even more accurate prediction results. Okay, so next I'm going to go into the next approach called metadata normalization, which is published last year in CVPR. So this work was also driven by student Bandy Lu and my colleague Aasa. So the motivation of designing this metadata normalization is that there are several problems we observe for the CFNet, or for the approach I just talked about in previous slides. And the first problem is that the CFNet can only correct features extracted at a certain layer, and it can only correct for one confounding variable at a time. So if you want to correct for a lot of features at multiple layer positions, you basically have to introduce more adversarial networks, and this will inevitably increase the computational burden. And lastly, of course, is you have this adversarial training, which involves this min-max of competing objective, which can sometimes lead to unstable convergence of your algorithm. So basically, we ask ourselves, what if we want something more lightweighted and can be inserted anywhere in the network at multiple positions, at multiple layers, to correct for multiple confounding variables. And the thing is that we do not want to add any additional computational complexity and want to also avoid the hassle of adversarial training. So we came up with this strategy of metadata normalization, or in short, MDN, which is essentially a non-trainable layer operation, which is pretty much similar like the batch normalization layer. And here we call it metadata because the work was first exposed to the CVPR community, not limited to the medical community. And metadata is more overarching term that refers to data that are not neither input nor output of a model. But in this talk, I will interchangeably use confounders and metadata to refer to the same thing. Okay, so before talking about the MDN operation, let's first revisit the concept of using general linear models, or GLM, to remove confounding effects. So let's say that you have a feature extracted at a certain layer, and how would you correct the confounding effects in this feature using GLM? what you do is that you can collect that feature across the entire population, and also can look at the confounder values of that population, and then you want to fit a general linear model regression, a GLM regression between the two variables to find this linear relationship between your confounders and your features. And once you do that, you can subtract the component related to confounder from the features, so that the residual component corresponds to the component in your features that are not related to the confounders. And the trick we realize here is that this entire residualization procedure can be written as a simple linear operation. And more importantly, this linear operator, this P matrix, does not depend on the input F. So it's purely based on the confounder information, which you know beforehand. In other words, regardless of what input features look like, the P matrix is fixed and can be predetermined. And all you need to do is to do this matrix product to gather residualized features, which you can then send them to the next layer as confounder-free features. So basically, we encapsulate this operation into this layer operation, and there's no trainable parameters, by the way, here. So you give the measures, you give the features F, MDN will just residualize the features with respect to the confounders. And another way to understand this operation is that with all the confounding information you know beforehand, you can pre-compute this subspace spanned by the confounding variables. And then once you feed in a feature F, we can just project it onto this confounder subspace, and then take the perpendicular residual component as the confounder-free component as the input to the next layer. So far the idea works really nice. The only issue right now is that we assume that we can collect this feature F over the entire population to build that GLM model. But in a deep learning setting, everything is batch level. So in other words, you only get a few samples at a time. You can never observe the entire population at once. So the trick here is that we proposed an approximated way of computing that projection matrix P using only data from a mini batch. And I won't go into details of the mathematics, but basically we made MDN operation a batch level operation in order to adapt to most deep learning settings. Okay, so here I'm gonna give you a simple exemplar application in a neuroimaging study, but we actually apply this approach to generic computer vision tasks such as face image recognition and also scene object detection. But specific to this neuroimaging study, where we aim to perform a multi-label classification between healthy controls, mild cognitive impairment, HIV, and HIV-associated neurocognitive disorder. We apply this MDN to correct for the site effect, because these data are collected from 3 different sites, and you know that imaging protocols can make a huge difference on the appearance of the image. So we need to correct for the side effects in making this diagnosis classification. And the results showed that MDN actually achieved really compelling classification accuracy compared to several other layer normalization methods. And again, if you look at the feature space, MDN can successfully remove the side effect compared to, for example, batch normalization. And lastly, I want to mention that we have our latest work on MDN published in NICA this year. And this work basically aimed to relax the constraint of batch level computation of MDN. And if you're interested, you're all welcome to take a look at this latest work. And at this point, I think I'm done with the 2 approaches that I want to talk about today. But I do want to make a final remark that, well, deep learning basically, we know that it gets into every field of science, and in my talk also including neuroscience and neuroimaging studies. So we should clearly recognize the pros and cons of deep learning methods. Apparently, deep learning has better sensitivity in identifying fine-grained cues in data related to the disease or whatever factors they're interested in. But the side effect is that it also gets better at exploiting those spurious relationships caused by confounding variables. So the conclusion is that accurately modeling of the confounders is very essential in analyzing medical or neuroimaging data. Okay, so I'll just stop here. I guess I'm ready to engage in some discussion.
Thank you so much for the talk. So we can go. We have some time for questions. There is already one question that came up in the chat earlier that I was also wondering about. So you mentioned a few times about, just feeding raw imaging data into deep learning models. But is there no longer some type of pipeline pre-processing just to improve the image quality? So perhaps Melanie, the one who asked a more specific question about this, I'm not sure if you want to unmute yourself, or otherwise I can read it out.
Yeah, I can ask the question myself.
Perfect.
So Qingyu, yeah, thank you so much for the super interesting talk. I'm also in computational neuroscience, so I highly appreciate this. So one of the things that was a little bit hidden on your slide 4 with the introduction of the deep learning is, of course, that we're not feeding a raw image in, we're feeding an image of a brain in, where we have extracted the brain from the skull and normalized it usually to a template. And somehow, do you think there is an issue with bias processing stuck in those steps in extracting the brain and in the usage of templates?
Right, that's a good question. I guess, yes, you're right that I kind of use this overarching statement of using the raw MRI scans as the input to the model. But in practice, you do want to first perform a skull stripping, or potentially doing some alignment of the skull strips brains to a common template in order for the machine learning model, deep learning model to work properly. I guess what I mean more precisely, what I mean by raw MR scans are those skull strip denoised, biofield corrected, registered brain. So that's precisely what I mean. And I guess the difference between deep learning and machine learning is that the follow-up processing steps in terms of tissue segmentation and brain region parcellation can be omitted in the deep learning workflows. I guess I hope that answers your first part of the question. And regarding the second part of the question, whether there will be bias also in the very first pre-processing steps involving skull stripping, yes, I believe so. The number one factor again is the side effect, because apparently the algorithm will work slightly different in skull stripping and biofield correction denoising, depending on which scanner and which site you get the image from. In this particular talk, I didn't design methods for those type of pre-processing steps, but I believe there are substantial strategy. I mean, some strategies proposed in those pre-processing steps in terms of correcting side effect.
Okay, thank you. We still have some time. There's a question in the chat. Do you see any advantage of the CFNet over the MDN approach?
Yes, that's a good question. I guess they both have their own advantages. So the thing about MDN is that it's really lightweight, closed form operation that doesn't involve in any additional network training. But the limitation is that it uses a simple linear or traditional linear model, regression model to find the relationship between features and confounders. That means that you basically have to put many of these small linear models at a lot of places to potentially remove any nonlinear relationship between confounders and features. So the strength of CFNet would be because you're using a neural network as an adversarial component to remove your confounding effect, you can directly model the potentially nonlinear confounding effects in your features.
So maybe a follow-up on that. Would it be correct to say that for MDN, you are learning slightly different distributions for a given set of metadata?
Well, the difference is really about whether you assume that you have linear confounding effects or nonlinear confounding effects in the features. So if you have nonlinear confounding effects in the features and you're only using a linear MDN to remove the effect, can you imagine that even though you residualize the confounding effect, you will have some remaining confounding component in your feature still confounded. And then what you do is that essentially you have to plug in this MDN approach at multiple places, and hopefully step by step, the nonlinear effects can be gradually removed in your network architecture.
Okay, thank you. I'm sure we can also follow up some more in the chat. Maybe I'll take opportunity to ask a question myself as well. So I was curious. So my background is not in neuroimaging, but I have seen, it was maybe 10 years ago that it seemed quite popular for prediction of AD to use diagnosis like AD, MCI in a multitask learning setting, actually together with age.
Right.
So then learning kind of a feature representation that is general for degradation, and then some specific layers on top of that, or perhaps it wasn't deep learning maybe, and then some specific features or weights that would be either only for age or either only for the diagnosis. Do you think those results in light of what we know now, is that, are you familiar with this kind of work?
That's quite relevant to the topics we're discussing here. So the thing about Alzheimer's is that, and also in general, neurodegenerative disease and cognitive impairment is that for this type of diseases, it's highly entangled with normal aging pattern. It's an abnormal aging or accelerated aging, and you want accelerated aging, and you want to separate that accelerated part from the normal aging process. But the disease and the normal aging lies on the same spectrum or continuum, so to say. So it's kind of a very challenging problem. And multitask learning is one way, I believe, the work you referred to disentangle normal aging part with the disease part, because you are simultaneously try to predict the subject age and simultaneously try to predict the disease label. And the way we did that is in our lab, in our own work, is to explicitly use the concept of disentanglement in the feature space to separate the normal aging process with the accelerated aging process. In fact, deconfounding by disentanglement in the feature space is another possible topic. I was going to talk about that in this talk, but due to the time limit, I actually omitted those slides. But I'm happy to send the relevant work in this chat later on.
Yes, that would be excellent. I'm very interested to follow up on this. So thank you so much again. Let's speaker. I think now we can move on to the next part of the program, and I'll give the floor over back to Enzo.